Hello, Starshines. We're going to go over day 3, unit 7, genetics. While we're going over this worksheet, make sure that you have your PowerPoint open so that you can follow along. Take your notes and also write down any questions that you may have. During the next live stream or the Zoom conference, we will go over any questions you have and the entire worksheet. All right, let's get into it. Aim. How does DNA transcribe its information outside the nucleus? Okay, so first and foremost, I'm going to definitely annotate nucleus and DNA. I know that DNA contains my genetic information, and I know that genetic information is passed down from my parents. And also, I'm going to write down for nucleus that that is an organelle in which we can find the DNA. Okay, so it's the DNA that holds the DNA. Now, for the do now, it's asking you to complete the complementary DNA strands to the following DNA sequences. So they gave us two. With the first one here, we can see that we have AAG, TCT, and GCA. If you notice, I kind of read it in groups of 3, and I definitely did that on purpose. I read it in codon, okay? So if I were to read number 2 as a codon, in codon, I would read it as CGT, AGT, and CA because there is not a third base there. What we're going to do next is find each base pair per codon. So in my first codon, I have AAG. A stands for adenine, and I know what my base pairing rule that adenine pairs with thymine, so I'm going to place a T there. Okay, I'm going to do the same thing here, and I know that G pairs with C, which is cytosine, so I'm going to write that there. Okay, and I'm going to just follow that model for the rest of the do now. Please take a moment and pause the video and complete the rest of the do now on your own. Press play when you're ready to go over it and check your answer. Okay, now for the rest of the first sequence, you should have had AGA, CGT. And for the second sequence, you should have had GCA, TCA, and GT. Notice the last 2 bases are not a codon because there is only 2. A codon is every 3 bases in a DNA sequence. Okay? Every 3 bases. Let's write this down. Codon equals 3 bases. DNA consists of 2 molecules that are arranged into a ladder-like structure called a double helix. A molecule of DNA is made up of millions of tiny subunits called nucleotides. Number 1, what does a nucleotide consist of? If you guys recall from yesterday's live and as well from the prior day's worksheet, a nucleotide consists of a sugar, a phosphate, and a base. Okay? So I'll write that down. A sugar, phosphate, and a base. And by base, I mean either adenine or thymine or guanine and cytosine. Number 2, draw a nucleotide and label each component. Now, as we read earlier, DNA is arranged into a ladder-like structure. So when I am creating or drawing my nucleotide, I'm going to keep that in mind. Now first I'm going to start with a circle for my sugar. Then I'm going to create the pentagon for the phosphate. Then I'm going to connect a base. Now I'm going to create a little receptor so that it can pair with its base. And for this base, I'll use guanine. Okay, so this is how we construct a nucleotide. And if we were to create the complementary nucleotide to that, it would look as such. I'm starting backwards, but it's okay. All right, so I have my, oops, hold on. I have my, because it's going to go in, I'll do that. And then I have my phosphate, and then I have my sugar. And as we know, G goes with C, so we're going to have C there. And as you can see, in drawing the complementary strand, I also made the backbone anti-parallel, because that is how they run when they are being duplicated. Alrighty. So here we have the backbone, and here we have our nucleic bases. Number 3, what are the 4 bases and how does the base pair rule apply to them? Now, if you can remember, sorry, it's getting late, there's an acronym that will help you to recall each base, okay? And that acronym is All Teachers Go Crazy. Now, if you notice within this acronym, we have 4 letters, and those 4 letters represent each base. The A obviously goes for adenine, the T with thymine, the G with guanine, and the C with cytosine. Number 4, what is a codon? As we spoke about earlier, a codon is every 3 bases in a DNA sequence. Part B, DNA notes. DNA has the information to build proteins. So essentially, when you think of DNA, think of it as being the instructional manual for our lives, for us, right? How we're here, how we function. DNA has all of the instructions. So how do proteins work? Proteins run living organisms, okay? As we know, enzymes are proteins, and enzymes control all of our reactions in our living organisms. And for structure, all living organisms are built out of proteins. And if you even think about the cell membrane also has protein channels. So proteins are everywhere, and they're very functional and vital for our living. DNA is mostly found in the nucleus, with the exception that the mitochondria and the chloroplast have their own DNA, right? As we spoke about that earlier, or on live stream, the mitochondrial DNA comes from the mother. Alrighty. The genes are instructions for making proteins, and it wants to be kept there. It wants to be protected in the lock. The reason why we want to protect the DNA is because we want to protect it from becoming damaged. If DNA is damaged, then we have ruined instructions. And if we have ruined instructions, we may not function properly. Proteins. Chains of amino acids made by a protein factory in the cytoplasm, and the protein factory equals the ribosomes, okay? So again, a monomer is just another word for building block. Amino acids are the building blocks for protein. Passing on DNA information. Now, as we already know, DNA is found inside of the nucleus. But in order for proteins to be made, they're made in the ribosomes, and the ribosomes are found in the cytoplasm. Now, we know that DNA cannot leave the nucleus because we don't want to risk it getting damaged. So what we're going to do is similar to DNA replication, we're going to use an enzyme. We're going to cut that DNA open, and we're going to make a copy strand, kind of like a photocopy, right? So we're going to make a copy of the DNA, and that copy is going to be called mRNA, or messenger RNA. This process is going to be called transcription. Transcription means using DNA to make mRNA, messenger RNA. A DNA strand template is used to make mRNA, okay? Now, because this is a copy, it's going to be slightly different than real DNA. Instead of having T, we now have U. So there is a new base pair if you look here. So here we have a new base pair, U and A. The U now stands for uracil, okay? G and C are still the same. The base pairs G and C are still the same. Note, uracil is only found in RNA molecules, okay? So uracil is a copy that's only found in RNA molecules. We will never use uracil inside of DNA. When you think of uracil, just think of, well, whenever you think of uracil, you should always think of RNA or mRNA. And when you think of mRNA, just know that that's a copy. It's a copy of a DNA strand, okay? In mRNA, there are no T bases, so U will pair with A. Okay, so RNA polymerase is the enzyme that is used to make mRNA. All right, so that's the enzyme that is used to unzip the DNA to make the copy strand. So here we're going to actually transcribe a DNA sequence. So here we have our first DNA sequence, and I'm going to actually write OG under it because it's our original strand. It's our template strand. Now, being that we're transcribing, our resulting strand is going to be mRNA. Now, we must remember, first of all, mRNA is a copy, and we must remember that with mRNA, there is no thymine. We have uracil. So that means that uracil is going to pair with adenine. There is no more T. So T by. Okay, so our sample DNA strand, our first codon should read as this. We have a T, so I'm going to put A. We have an A, so I'm going to put U. We have a C, and I'm going to put G because that is the normal base pairing rule for cytosine. Now, pause the video and try the rest of the transcription for yourself. The remainder of your strand should read as the following: CGU, GUA, and AUG. Okay, so genes and proteins. Now that it has been shown that DNA is what makes up the genetic material, it is time to look more closely at genes. What is a gene? A gene is a specific segment along a DNA molecule, made up of linear sequences of nucleotide subunits. It is, in effect, a package of information that tells a cell how to make proteins. Likewise, proteins are polymers made up of linear sequences of amino acid subunits. There are 20 different types of amino acids. The order in which they assemble determines the type of protein that is made. That is important. We will learn more about this tomorrow. This sequence determines the shape of a protein molecule. It is the shape of a protein that allows the molecule to do its work in the cell. Cells have to use the linear sequence of subunits in DNA to build amino acids for protein. Yet, in all cells except for bacteria, DNA is stored in the nucleus, and protein synthesis occurs outside the nuclear membrane at the ribosomes. These small organelles are distributed throughout the cytoplasm. How does the genetic information contained in DNA within the nucleus get out of the ribosomes? A third type of molecule called ribonucleic acid, or RNA. It works as a helper to transmit the information. That is, the genetic information flows from the DNA to the RNA to the protein. From DNA to RNA, each gene is a portion of a chromosome, in effect, a portion of the DNA chain. An RNA molecule called messenger RNA does the job of moving the information in the linear base sequence out to the ribosomes. DNA is copied into RNA in a process called transcription, which is similar to DNA replication. First, the DNA double helix opens up where a particular gene is located. Special enzymes begin to match up RNA subunits with the corrected DNA subunits. The RNA molecule has the same base sequence as one strand of the original DNA, except in RNA the base uracil substitutes DNA base thymine. This RNA molecule then goes out of the nucleus through the pores in the nuclear membrane to the ribosomes in the cytoplasm.